Welcome everyone to our first online lecture in human physiology. Anatomy 36 is continuing online, and we have two more topics to cover before we take our first unit test. And our first topic will be cell division, mitosis, meiosis, and then I also have a little section on cancer in there. And then we have one more topic on cell communication. But let's get started first with mitosis. So the full life cycle of a cell is also known as the cell cycle. Most of the cell's life is spent in something called interphase, and that is the part where the cell is not worried about dividing. Then a much shorter portion of the cell cycle is mitosis, and that's what we'll be focusing on. But let's take a look at the full cell cycle. So the interphase portion of the cell cycle is divided up into G1, a growth phase basically, and that's where the cell produces a lot of messenger RNA proteins, just goes about its business. It will just work for the organism that it's in. And most cells basically exit the cell cycle or the continuation of the cell cycle right there and go into something called G0 arrest. The next part of the cell cycle is called the S phase. S is relatively easy to remember when you think of synthesis of DNA. This stage is when the cell copies its entire genome in preparation for cell division. Remember, a cell only has one nucleus, and so if you want to divide, then you need to make sure that the two daughter cells will each receive a full set of genetic information. So therefore, we need to copy that at some point and make out of each chromosome with one sister chromatid, we need to make two sister chromatid chromosomes out of them. So that happens in S phase. And then we have sort of a final growth spurt in G2. That is the stage just before we go into mitosis. And at that point, the cell needs to grow, do some final steps, and it's at that point committed to go into mitosis. So here, the same information one more time in a diagram. The cell cycle can be divided into interphase, which is all the yellow stuff right here. So from here all the way around, all the yellow stuff to here, that's all interphase starting here. And then mitosis is the actual cell division part of the cell cycle. Interphase starts out with G1, that's a growth stage. Most cells will stay indefinitely in this stage. You kind of exit the cell cycle and go something into that's called G0 arrest. And they just do what they're supposed to do and do a good job for you. And once it's time to move on in the cell cycle, they might return into moving on here. So they exit G0 and then move into S phase. And S phase, that's DNA replication. So we need to make a second copy of the entire genome in order to do cell division. So basically the chromosomes, they go from a stage where they have one sister chromatid. Oops, this one does not look very good, but anyway. And then they go into these more typical X-shaped structures that look like, oh, this is terrible. Anyway, so oops, let me move it from here. We can see it. So one sister chromatid chromosome look like this, and then we're going into these kind of chromosomes. They look like this. And once that has been accomplished, then we're going into G2, which is another growth stage. And once that's happened, then the cell's undergoing mitosis. The molecules that are driving the cell in the cell cycle, there are many of them, but the most important class here are called the cyclins. And since I have a little discussion on cancer in here, I just want to mention this to you that these cyclins, they're basically the driving force that tells the cell to move on in the cell cycle. There's a whole family of proteins. Here's one mentioned, cyclin D, but there's various others. It's a whole family. And if they, for some reason, mutate and they will not turn off anymore, then what happens is they turn into what's called oncogenes. Oncogenes are mutated forms of normal genes that promote a cell to turn into a cancerous type of cell, so from normal to cancerous. We also have what's called tumor suppressor genes, and if they are mutated, it's just as bad. You can compare oncogenes and tumor suppressor genes basically as the oncogenes as a stuck accelerator in a car. So you're gonna run every red light if your accelerator is stuck. And the same is true for the cell cycle. The cell just won't stop the cell cycle if there's an oncogene present. And tumor suppressor genes could be compared to a failing brake in a car, where, well, again, if your brake is failing, then you're going to run every red light as well. So tumor suppressor genes, if they mutate, it works just like a failing brake in a car. They are normally supposed to inhibit cancer development, but if they fail, if they're mutated, then it doesn't work anymore. So one very famous tumor suppressor gene is p53. That one, you may have heard of that. Some people discuss this one. And then there's another important tumor suppressor gene. It's p21. Anyway, so we'll come back to this idea here with the cyclins when we talk about cancer. So coming back to mitosis, so we talked about the cell cycle as a whole, and a relatively short segment of a cell's life cycle is the actual cell division. Now, in eukaryotic cells, you have a problem because there's only one nucleus, and that nucleus, the two resulting cells both need a nucleus. So we really, the steps of mitosis revolve around what's happening with that nucleus. There are 4 stages in mitosis: prophase, metaphase, anaphase, and telophase. Roughly, what happens during these stages is as follows. In prophase, think of P as the cell the nuclear material becomes plain to see. So what we first need to do is condense the chromosomes, the genetic material, to more manageable packets. The chromosomes therefore become visible, and you think of prophase, the chromosomes become plain to see maybe. In metaphase, those condensed chromosomes, they line up in the middle. So metaphase, M for middle. These chromosomes now line up single file along this, what they call equatorial plate, or the middle of the cell. Once that has been accomplished, then we need to split the chromosomes that have 2 sister chromatids. We need to split them apart so that each new cell is going to have 1 sister chromatid each, and that happens during anaphase. So anaphase A for apart the chromosomes, the sister chromatids, they will split apart and get pulled to opposite poles. So the 2 sister chromatids go to opposite poles. And in telophase, telo T for 2 new cells, we're going to have 2 new cells, 2 resulting daughter cells in this final cell division step. So here the same information on another slide. We're starting out with interphase. And the interphase is characterized that you really don't see much in the nucleus. There are no distinct structures in there. It's just all one fairly homogeneous material. So the nucleus is visible, but you don't really see distinct structures in there. Once we enter prophase, the chromosomes become plain to see. And you can kind of see here in this slide that the chromosomes starting to become distinct structures. They are very condensed at that point already, and they starting also the nuclear envelope is starting to fall apart. Moving on to metaphase. In metaphase, you can see that the chromosomes, they are at their most condensed state at that point. They all look like little X-shaped structures, and they all have 2 sister chromatids that's held together in the center by a centromere. In metaphase, that's the stage where they're lining up single file. So each one of these X-shaped structures, you're lining them up right here in this equatorial plane right there. You also have poles that have formed, these centrioles, and they are reaching out with spindle microtubules toward each one of these chromosomes from opposite sides of the cell. In anaphase, these spindle microtubules have attached to each chromosome, and they start to pull the 2 sister chromatids in opposite directions. So each one of these chromosomes has now 1 sister chromatid. And then the final step is telophase, and the actual division of the cell into two new ones. That's called cytokinesis. So in telophase, basically the sister chromatids, they have arrived in opposite poles, and now we need to start forming new nuclear envelope around the genetic material. So that means the new nuclear membrane is formed. The nucleoli, they will reappear, and then cytokinesis is the final step that completes the cell division here. So kinesiology, you know, that's movement. So cytokinesis is basically the movement. The cell gets really long and it starts to pinch into two new ones. It's a little bit different whether it's an animal cell or a plant cell. Since we're here in human physiology, we're going to take a look at what it looks like in an animal cell. So here in this image, you can see that the cell gets pretty long. And what happens to an animal cell is that you're going to have this cleavage furrow. It's basically like a belt that forms around the cell and tightens up more and more and more, and then eventually pinches the cell into two new ones. Or up here you can see it is a stained cell under light microscope. Yeah, we're almost done already with mitosis. Just a few words about the control of cell division. People might wonder, what is it? When does a cell know when it's time to divide? So there are, of course, chemical signals, such as these cyclins and growth factors, hormones that can come in that promote the cell to go into cell division, to go into mitosis. Also, it is thought that the surface to volume ratio of cells is important. We talked about this in the lab. It's very important that cells stay small so that they have a large surface area compared to a small volume. And that is because they want to take advantage of diffusion. And if the volume was too big, then diffusion would not be something that would be possible. So cells have a desire to stay small so that they have a large surface area relative to a small volume, and when they get too big, that's basically a signal for them to divide. Then there's another thing called contact inhibition. That is all normal cells have that. So if a cell in the human body that's normal has a neighbor on all sides, then that is a signal to not divide. So in other words, if your skin is unbroken, then there's no need for these cells to grow extra tissue. Now, if you have, let's say you burned yourself or you have an injury on the skin and now you need to grow a new patch of skin, then there is a need for these cells to divide and create and sort of heal the tissue. So the idea of contact inhibition is when everything is fine and the tissue is intact in an adult organism, then you really only need to replace what has died or has been damaged. And then we already mentioned the cyclin-dependent kinase complexes, these CDKs, that's what they're called, cyclin-dependent kinases. These are chemical signals. They've called in this category chemical signals that strongly promote the cell to move on in the cell cycle. Here we have another aspect, and that would be the role of telomeres in cell division. Now, telomeres are the ends of linear chromosomes, and if you remember this from our lecture on replication, these ends of linear chromosomes, they shorten with every round of cell replication. So we're losing about, 50 to 100 base pairs, and that shortening of these linear chromosomes seems to contribute to cell aging, cell death, and also aging of the entire organism. And finally, cell death here. There are a few ways that cells can die. Pathologically, of course, if the cell gets sick or something happens to it, gets injured, then we call that type of cell death, that would be called necrosis right here. The cell will burst usually and then just disappear. Then there is something called programmed cell death, apoptosis and programmed cell death, basically the same thing right here. So this means that the cell, there's something wrong with it, and it gets strong signals for it to die. And these signals are mediated by enzymes. They're called capsases. These are execution enzymes that basically tell the cell, hey, it's time to go. You are abnormal. Something's wrong with you. You need to die for the good of the entire organism.